Welcome to the daily recap of day 3, EULAR 2022. We are all here in Copenhagen, unlike day 1, day 2, where we had sort of all over the globe people. All 4 of us are here enjoying the overcast, rainy weather. Skies are getting clear tonight. Maybe we'll have some sun tomorrow. But it's good to see everyone face-to-face at a meeting. We've had a good time together. We're going to discuss our highlights from day 3 here at EULAR. I'm going to ask my friends and colleagues here to introduce themselves. Let's start with Janet Pope.
I welcome everybody to Room Now, our daily recap. Janet Pope, I'm a rheumatologist in Canada, and I'm at Janet Burdope is my Twitter handle.
Yes.
Hello, my name is Yusof. I'm a rheumatologist and a senior research fellow at Leeds. So my Twitter handle is Yus6Yusof.
And Orly.
Hi everyone, I'm Aurelie Najm. I'm a rheumatologist in Glasgow and a senior research fellow. And my Twitter handle is Aurelie Remo.
Excellent. Okay, so let's start with Orly, and we're going to give you our favorite abstracts of the day and discuss them a little bit. Go ahead, Orly.
Thank you. So I got quite excited today by the oral presentation 0258, which was actually talking about isokibep in PSA. So it's not a new mechanism of action. It's an IL-17 selective inhibitor. However, what's quite interesting with this molecule is that it's a fusion protein that is actually much smaller than the monoclonal antibodies. And we know that monoclonal antibodies can sometimes, the way it binds on the tissue and the way it actually penetrates and access to the tissue, it's not always optimal. So they try to bypass these issues by making a much smaller molecule. And today, what we presented was actually the results of these in a phase 2 trial in PSA versus placebo. So the ACR 50 at week 16 was actually about 52% responders, which is actually good, which is pretty close to what we already know. In the PASI 90 was about 50%. But what got quite exciting was the result on enthesitis. So the Leeds enthesitis score in only one location was improved in 88% of the patients, which is actually quite much better than what we usually see. And this could be actually linked to the fact that it penetrates the enthesis much more than the monoclonal antibody can do. So obviously, it's a phase 2 trial. It's only a few patients. So I would be very curious to see what it's going to be in the phase 3 and how it's gonna behave with enthesitis. But I thought it was quite exciting, and that's why I thought it would be good to share that with you.
Yeah, this is a novel compound. You know, just on one hand, you could say it's yet another IL-17 thrown into the mix, but being a dual IL-17A, IL-17F inhibitor, maybe it is different. And I can say that it is different from the psoriasis trials, where the efficacy looks different, but also the safety looks different. While you can theoretically get candidiasis, we don't really see any candidiasis with any of the IL-17 inhibitors, really. But with these dual inhibitors, the clinical trials have shown, 8 to 15%, and it's oral candidiasis. It's not like any major invasive candidiasis. So there is something mechanistically different here. And I think that can also end up having a positive efficacy signal. But what do the rest of you think about this, this new compound as it enters the later stages of development? Janet?
Well, I think that it's got similarities. Of course, it is in the class, but it seems a bit different. I agree. And if it's different, maybe it's going to have safety signals that are different, maybe not. But the efficacy is in line, if you ask me, with the other ones.
Yeah. Yus, what do you think?
Yeah, I think because there's quite many options now in psoriatic arthritis and SPA areas. I think it's important to see, like, what difference, features that they can distinguish from other treatments. I think that will be something interesting that we look forward, when they go into the bigger phase studies. Yeah.
Yeah, and I think Orly's point about that really high enthesitis response. What is interesting about most of the drugs in the SPA PSA space is they all seem to have kind of similar enthesitis, dactylitis kind of responses. Nothing really has jumped out. Maybe this will be an opportunity for one to jump out. What's your next best?
Yep. So yeah, in line with what Aurelie was saying just now, so this is a smaller study, but I think sometimes you need to start with a small number. So I think, so why don't we start with n equals to 5 patients? So it's been presented in oral abstract session today. The title is OP0279. It's 0279. So that's the abstract number. So this is a new potential new therapy in resistant and refractory lupus. So a therapy that have been used in cancer, hematological cancer, which is the CAR T-cell therapy. So Professor George Schatz's group in Germany, so what they've done, they have really resistant patients resistant to biologic, including rituximab and belimumab. So these patients, what they did is they took the T cells from the bloods, and after that they genetically engineered it to be armed with the chimeric antigen receptor, which is the CAR, and this is primed to attack the CD19, which is your B cell that driving the lupus. Interestingly, so what they did, so prior to the starting treatment, they've stopped all medication and they infused this treatment back to the patient. And then they even didn't not start any steroid or any other immunosuppressants. And majority of this, 5 patients actually were in remission up to 6 months. So I think it's really interesting. All 5 patients had resolved the SLEDAI score. Some of the B cells even not return at 6 months. And antibody as well. Half of the patients have no antibody double-stranded DNA at 6 months. So I think it's really interesting, and I think certainly need to be forward for randomized clinical trials for it to be definitive.
Were you convinced? I didn't see the presentation, but I read the data in that they started out with SLEDAIs of 8 to 16, and they had failed everything from mycophenolate, rituximab, cyclophosphamide, belimumab, hydroxychloroquine, everything. Were you convinced that these were really severe patients and really refractory? Because if they were, it's sort of shocking that their SLEDAIs went to basically 0 across the board.
Yeah, I think that's what they've portrayed, but it is n equals to 5, and it still need to be done a proper randomized trial. And as in the trial with lupus, always difficult. Although there's composite outcome measures, SRI-4, BICLA, a lot of trials failed, following positive phase 2, but then they fail in phase 3. So I think just have to wait and see.
One of the things about this therapy is that cytokine release syndrome that has been reported. Did they report any of that with these cases? Janet, what did you think of this report?
First of all, this is desperate measures require sometimes desperate measures and desperate treatment. But I think it shows us a way that changing the immunology and catering towards personalized medicine might really help people. On the other hand, this would be so expensive that it's unlikely to ever be ready for prime time other than our most severe patients. But I think it gives us an idea that if you're personalizing what's wrong with the cells, you might actually help people. So I think it gives us a hint. Biologically, I don't think it's going to happen in many centers, if any.
Yeah, it will be surprising. Orly, did you see this presentation?
No, I didn't see it, but it's quite impressive to see these results. It's kind of hard to believe, right? I would really like to know what's happening after 6 months as well, because I think I think it would be really interesting to know, if they actually cured, which is really hard to believe. But it's quite exciting.
Yeah, and the fact is that they all went off of all therapy, including steroids, after this, and that was a fairly long. But I got to tell you, I did some of the first biologic trials in RA back in, I want to say, like 1988, and we gave monoclonal antibodies, with immunotoxins, and when you give this kind of therapy, it's a tremendous shock to the immune system, and everything shuts down, and there's a lag period and whatnot. And so the data is always very exciting, but then when it gets put to the rigors of a clinical trial, things don't quite look quite so magical. But still, I think this is a very bold study that Yus has presented, that done by Dr. Georg Schett and his group. So kudos to them. Let's move on to another one. Janet, what was your one of your favorites from today?
Right. So not as dramatic today as what we've already heard, but I think it's a little clinical pearl. So I looked at a poster, and it was poster 0242. And it was basically looking at the safety of antimalarials when they're in combination with an advanced therapy. So they combine biologic and targeted synthetic DMARDs with hydroxychloroquine and compared it to those without. So first of all, there'd be a bias in hydroxychloroquine, are you doing it instead of, say, methotrexate because the patients are sick or have contraindications, or you're doing it in combo. So we don't know all that from the poster, but it's what's interesting. So they looked at two things to me of interest. The first thing is they looked at retention. And if you were on hydroxychloroquine, you had better retention of your advanced therapy than if you weren't. And we know from many old studies that retention, a lot of these were TNF studies from way back when, retention was better on methotrexate, then on leflunomide if you weren't on methotrexate, then on sulfasalazine. And they never really looked at hydroxychloroquine. We thought it was kind of weak. So this is in RA patients, and they're showing retention is better on than off. It's statistically significant. The curves aren't that different, but I thought that's a good take home because I do have people on antimalarials and often stop them when they're on an advanced therapy. The other thing of interest was they looked at serious adverse events, serious cardiovascular events and total cardiovascular events, and serious infections, as well as hyperglycemia and hyperlipidemia. Less hyperglycemia, less hyperlipidemia, that's been seen in antimalarials in RA and lupus. I believe it. It gives me face validity. Total infections, though, were less than those not on antimalarials. Interesting. And something that I wouldn't have expected, serious AEs were less. I would have thought they would be the same, but serious cardiovascular adverse events were 1. There was no protection. And that's where we see the protection in lupus. And we've seen it in RA in one of the Scandinavian studies before that, in a population study of RA patients in Scandinavia, there was actually, if you're on hydroxychloroquine, you had a decreased SMR, so less mortality, including cardiovascular. So cardiovascular didn't pop up. The other things did. I think more will come, but, increased retention for a drug that's pretty inexpensive, helping lipids, helping hyperglycemia, not bad.
Yeah, the other thing that was surprising in that was less hepatic toxicity, which I found, and of course, there has a mixture of biologics and targeted synthetics that the patients are on in addition to their hydroxychloroquine. But I wouldn't have expected that the hydroxychloroquine group would have a better hepatic profile for some reason.
And I'll tell you why, Jack, that I didn't mention it, because it was quite protective, meaning quite less. I wondered if there was a confounding, so I didn't tell you this part, because if you're on methotrexate and you can't use it because of hepatic things, you're not going to maybe worsen transaminitis. I don't know. It was kind of interesting, but I didn't know what to say about it, so I left it be. But yeah, they did find that.
Yeah, we didn't have all the data, but it was surprising that they didn't have a lowering of the cardiovascular event rate, which has been reported in several different ways, especially in a lupus population, also in an RA population as well. But, the more we practice, the more we learn. Everybody should be on hydroxychloroquine. What do you, the rest of you think?
I mean, if I think, go ahead, yes, go ahead.
Yeah, thank you, Orly. Yeah, so certainly because my field is lupus and connective tissue disease. So I encourage, all of my patients, to be on hydroxychloroquine. I don't really taper them, like later on in the life as well. So, and majority of my patients, they tolerate it well.
Yeah.
And this is looking at RA. I mean, we don't think of it as being that effective, but there's a good safety profile other than, after maybe a decade or two of use, your retinal toxicity does go up quite a bit in some people.
Yeah. Let me go on to what I thought was a big report from the day, and big in multiple ways, the Gloria Study presented by Dr. Martin Boers. This has been probably a 10-year project for him. This is a 2-year trial, pragmatic, double-blind, placebo-controlled trial where people over age 65 receive, in addition to their usual therapy for rheumatoid arthritis, receive either 5 milligrams of prednisolone or placebo. And it's a 2-year outcome study. And at the end of 2 years, he showed that the prednisolone group had basically less disease activity. They did better. I mean, there was separation of the lines in the sort of Kaplan-Meier plot, if you will, of outcomes. And there was less radiographic progression. But the differences here were at the edge of being significant. So they were probably statistically significant, but were they clinically meaningful? So the DAS-28 score was 0.37 less. And then in ideal patients, no dropouts, ideal compliance, whatever, it was as much as 0.62 on the DAS, reduction scale. Now, and again, as far as the X-rays, minus 1.7 units, again, that's a 400 unit scale. It's hard to imagine, but it was meaningful from a statistic standpoint. But was it really meaningful? It's hard to know. Now, the converse side is the amount of harm that was done. Harms were measured in many ways. A lot of them were adverse events related to steroids, but they had more adverse events overall, 24% more, with a relative risk of 1.24. That was significant. And most of that was taken up by infections, most being non-serious infections as opposed to serious. But there were more infections and more adverse events. The question is this a reasonable trade-off, especially in light of the EULAR guidelines presented by Dr. Smolin on Wednesday, where the EULAR guidelines said, yes, you should be using bridge therapy, and yes, you should be getting off of it within 1 year. And that's very different than ACR guidelines would say, no, don't use the steroids. Don't use the steroids unless you have to. And now we have this Gloria data saying it's okay if you're willing to get some benefit and maybe some risk. So how does the rest of the panel feel about this data? Does this help you in what you're doing? Orly, what do you think?
So this trial, I think, was presented as well at ICR, and I remember I had the chat with Jonathan Kay about this, and there's actually a record of us chatting about that, because see, that's the thing, I'm not convinced. If you ask me if I were tomorrow give steroids to my 65-plus-year-old patients, I probably wouldn't. I would probably keep on going, especially I think we have kind of a body of data yet saying that, biologics are not necessarily of a worse outcomes in terms of safety in patients that are above these ages. So I probably wouldn't change my practice based on these results. I don't know what the other thing, but
yeah. Yus, what do you think?
Yeah, so I'm not a fan of steroid. So I wouldn't really want to put my patients on 5 milligram. I mean, potentially, like, they also may have concurrent osteoarthritis. So they may feel okay with the steroid in the beginning, but later on down the line, once they start getting comorbidities and you trying to win them off the steroid, it will be really going to be so struggling, like, to get rid of the steroid because of the effect on osteoarthritis as well, potentially. So, I think, it's not going to be my practice. I don't think so.
Spoken like a true lupologist, where the thinking is maybe we should not use so much steroids in lupus, unless, of course, it's lupus nephritis when you've got to use steroids. Janet, what do you think? Are rheumatologists too in love with steroids?
Right. So I did talk to Martin Boers about this because I chaired the ACR poster tour when he had the late breaking, or at least the poster of it at ACR. And what I said to him, and he had a good answer, I'll tell you, but I said, you know, what if you just did, we just talked about hydroxychloroquine. What if it was methotrexate plus hydroxychloroquine? That'd be a safer option, still affordable, the whole bit, or triple therapy. And then you don't have to go on to an advanced therapy for cost, risk of infection, etc. And he basically said, well, it does follow the EULAR guidelines. There would be a proportion of patients in whom the difference would be highly clinically relevant, keep them functioning, right? Because a mean change, there would be some people getting a DAS, like a DAS of 0.6 deltas were pretty good. 0.3 is not unreasonable as a mean, because some people are doing better than that. So if it were a patient in many of the Canadian practices, we would do two things. We do our optimal dosing of methotrexate. Some would go double or triple, but some optimal dosing methotrexate, absolutely, with, you know, serious disease. But we would also probably do intra-articular or intramuscular glucocorticoids to get them going more quickly while the drugs take time to kick in, because we control the steroids. They're not taking it for their mechanical back pain, or as we heard, their OA or other things. And that way they're not on it long term, because I don't want osteoporotic fractures in my patients.
Yeah, I had a little bit of a debate with my friend and colleague, Catherine Dow, who's been tweeting. She did a Twitter poll basically asking, what do you Twitter rooms think about should we be going the ACR way, less steroids, no steroids, or the EULAR way, okay to use steroids? And then she's told me the Twitter poll was in favor of, we're okay still using steroids. So I think the jury is still out, and I think this is a moving target. Keep doing what you used to do. It only is going to get you what you've always gotten, and you're not going to get any better. So, but then what's the alternative to using steroids? Janet's given us a few. Let's move on. On a final round, this is Jeopardy round. Let's do sort of quick hits. Let's start with Yus.
Yep, so the quick hits, the OP0247. So this is a study, a very large study involving 1 million patients with IMID. So Professor Philip Mees presented in the COVID abstract sessions. So they were looking in, he used machine learning, trying to identify predictors of poor severe COVID outcome, including hospitalization and death. So, as you may be aware, pre-vaccination periods, so the data from registries, the Global Rheumatology Alliances showed that the JAK inhibitors and rituximab had an increased risk of this severe outcome. So what Philip Mees and colleagues have seen in this study, so it's they divided into two eras. So one is pre-vaccination and one is post-vaccination. So the pre-vaccination, they found that there was a weak signal of association between JAK inhibitors and rituximab with poor outcome. However, after vaccination, this weak signal has gone. So essentially, there's no biological DMARDs. None of it actually that were tested actually were not associated with poor severe outcomes, so indicating that the vaccinations work, and potentially the less severe variants that we're having now post Omicron as well.
Yeah, I think we get a variable signal. We get a consistent signal on steroids putting our patients at risk, on rituximab putting our patient at risk. We get a variable signal on JAKs. Not all studies have shown that. A few have. I think it reflects the severity of the patient. Patients who are severe active, no matter what therapy they're on, it's the severe active disease that puts them at higher risk for poor outcomes. Let's go on to a quick hit from Janet.
Okay, so this is just a wee honorable mention because it's a new way of thinking, but that not ready for prime time. So this was Chen et al. It was poster 0014. And what it was looking at was recalcitrant or not patients, adults with Still's disease, so systemic onset disease, Still's disease. And they looked at 7 patients and they treated them with either tofacitinib or baricitinib. So 5 with tofacitinib, 2 with baricitinib. So what's interesting, 4 had failed lots of prior treatments. And so you'd go, well, that makes sense. But 3 were de novo. So I'm not sure where that's coming from. And what they found was they looked at the usual biomarkers, looking at the CRP and the white count, the ESR, things like that. And they found most had a partial response. Some had complete response. And I'm not sure exactly how you modify or adjudicate partial versus complete response. So I thought this isn't ready for prime time, but if I have a recalcitrant patient who's been through steroids, adult ones are usually getting methotrexate, 1 or both of the IL-1s separately, serially, and IL-6 or 2 IL-6s, again, not together, then I don't know what to do. So I thought, well, maybe it's a good bailout, but I wasn't convinced that this was going to change my practice anytime soon, but kind of an interesting thought.
Yeah, Hughes and I covered Still's disease at ACR, and we covered, I think, a partial version of this report and found it interesting. It's interesting from the standpoint of that JAK inhibitors may have their best place in patients with MAS, especially after the hospital, after you treat them with, I mean, I love the emapalumab, the gamma interferon monoclonal antibody for really sick patients, but most people are getting either etoposide or cyclosporin, depending on who's doing the consulting, and then either an IL-1 or IL-6 inhibitor with that. But after discharge, what are you going to give them? Well, I've used the JAK inhibitors, and it stands to reason that they may have value. Of course, they're not inhibiting IL-1 or IL-6, and many of their effects are downstream and could be, if you listen to Peter Nagorovic yesterday, could be from mTOR and other things that rapamycin might have an effect on for MAS, but it's downstream of that. So nonetheless, it's encouraging, right? It's good to have another option for those patients who look really, really sick. So Orly, what's your quick one?
Yeah. So quickly, I wanted to talk to you about this new imaging modality. So it's obviously not something that's going to be implemented in clinical practice tomorrow, but I thought it was really interesting. So it's Andreas Ramming that presented its oral presentation 0256. So what they did, because, we know that in PSA, there's an activation of the mesenchymal compartment and especially fibroblasts. So what they did is that they made the fibroblast activation protein tracer based on the CT scan. And so what you can do is you can actually see fibroblast activation through imaging. And so they showed really nice images where you could see how well the tracer uptake was correlated with pain, with inflammation in peripheral joints, but also, and that was quite interesting, at the enthesitis level in the spine, you could see some uptake as well. And what was actually really nice is that the uptake was predictive of structural damage 3 to 6 months later with MRI. But also, the other cool thing was that it was quite sensitive to change. So they showed images of sacroiliitis, and you see from month 0 where it's flashing to month 3 after treatment, it's completely gone the signal, which we don't have with MRI or any of these modalities that take a very long time to change. So, I mean, obviously we're not going to use that tomorrow, but I thought that was really interesting for once to have an imaging modality that looks into the cellular activation.
That is cool, and coupled with CT, and given the fact that it shows up in entheses, that might be something to look for in the future. My last one is going to be on the follow-up of the ARIA study. The ARIA study was an abatacept in preclinical RA presented last year, 100 patients, 98 evaluable. They showed us the 6-month data at ACR showing that compared to placebo, those who received abatacept at 6 months had less MR inflammation, had less progression to RA and whatnot. At that time point, they stopped the active intervention being abatacept and then followed people for another 12 months off therapy. And the question is what was going to happen to them. Now looking out at the end of 18 months, there's less progression to RA, 35% with ABA and versus 57% on placebo. This is, in my opinion, the first study to show actual prevention of RA. You might say it's actually a stalling of development of RA because you have to follow them out further. The number needed to treat here is 8, which is not bad. but if you don't like this data, that makes some people crazy. It's like, I'm not going to give, 8 people an expensive drug with potential toxicity when they don't yet have rheumatoid arthritis with the hope that they won't get rheumatoid arthritis. And so this could go either way, but this is of a number of studies that have been done in this area. It's the first one to show the potential for prevention. I'm still not going to treat patients with preclinical RA. I'm only going to treat what they have. If they have synovitis, I'll treat that with something, and I'm not sure what. But arthralgias and a positive CCP, I'm just going to follow you every 6 months. Does this data change any of your opinion about what you would do? Janet's nodding no.
Yeah, I think it changes the way I think biologically. I think that's really like it's a hint into future unlocking of who will get it. But I think there's, as I say, a bit of a number needed to harm, not the safety or lack thereof of the drug, but the number needed to harm is we're treating 7. And what's the end game on 7 to help 1? So 1 out of 8 gets help. We're doing an expensive thing, might mess up their insurance, et cetera. And we don't know that at the beginning of clinically relevant RA, that in many people, not all, but in many people, we can turn it off for a while because they're a captive audience. And you'd see them every 6, as you say, but you'd say, you call me if you see swelling, you're stiff for more than an hour. You give them guidelines. Maybe you'd have them do a lab every 3 months for a bit to see inflammation. So I think captive audience you'd follow, but I wouldn't start this right now, but I love the idea of it.
Yeah. All right, folks, let's adjourn and get back to our preparations for day 4, which is tomorrow. I'll remind the audience that we will have a day 4 report. It's just not going to appear on Saturday tomorrow. We're going to record it after the meeting and probably present it to the audience sometime early next week, like Monday or Tuesday. Watch for it on the live stream that we have on our website, on YouTube, Twitter, Facebook, and LinkedIn. Thanks so much, folks. Have a great night. Appreciate all the input.